trying different technologies for single cells to see which one is really the best one for us. Also, related to the last question, actually, you never can get all the population of cells from a bulk seq because you lose some of the expression. Statistically, you lose some of the expression if it's very low. So that's why the systems biology approach was designed. And by far, this is the easiest and more affordable technology we've used. And we are trying all of them, almost. So I'm showing data with the version 1 kit, which is released a couple of years ago. We were a little bit slow to introduce it because we're building our research infrastructure, and part of that is our platform for genomics and transcriptomics. All of our platform is based on long-read nanopore sequencing due to some of the advantages it has, but it has also, it was very noisy before, and only this year there is a model that has accuracy of 30 now, but so far it was very challenging. So what I'm going to show you is a simplified approach, not to a whole tissue. We have one project for several years. We used the long-read direct RNA sequencing to seek for long non-coding RNAs and other RNAs that we can analyze directly. So we enrich one C cell line PC3, which is a prostate cancer metastatic cell line. It's bone marrow metastatic line. Through a cultivation protocol, we enrich this for stemness, stem cell markers, and we validate this and also saw these cells with for CD44 and CD24. Pathologists use also CD44 as a marker, but truth to be told, a lot of these CD44 isoforms are very specific, and only some of them are actually specific for metastatic disease and some of them for stemness. So CD44 is not one simple molecule. Therefore, we need transcriptomics. So we started with RNA sequencing. What we found, these figures on the bottom, these are correlations between RNA pool, long non-coding RNAs with poly-A, and we had one method to tail the non-poly-A tails, long non-coding RNAs. So when we compared with very deep sequenced PC3 cell line on Illumina, there are still some molecules that Illumina didn't detect. So this is actually where we get our interest. As you see, the CD44, the wild type adherent cell line, compared to the stem cell enriched cell line, they have different isoforms. So there is an isoform switch when you actually promote the stemness. So why long-read nanopore sequencing? One of the benefits is the full-length transcript identification and isoform detection. Novel fusion phenomena detection, because sometimes you directly sequence the actual molecule. You do not gather this information through bioinformatics, you just sequence the real molecule. Also, cryptic zone detection is easier with this kind of method because it sequences longer piece of RNA directly or indirectly. And the ability to make a deeper whole transcriptome characterization. Also, the new emerging possibilities for rapid transcriptome changes in bulk-seq. Unfortunately, not in single cell, but future will tell. And easier identification and clearing of artifacts, combining cDNA with direct RNA bulk-seq. So single cell transcriptomics is the next step when you want to analyze a complex sample. In this case, you'll see that we analyze only a single cell line, not a multitude of cells, and yet we have more data due to the single cell sequencing. So we used the GScope version 1 kit. My colleagues used 2 chips, one for the adherent and one for the stem cell line. We prepared libraries and we sequenced them on the grid ion device with regular mini ion flow cells. What you get is a POT5 file. This is the new file system that is produced by long-read sequencer. And this data set, it is base code with Dorado version 1 from this year with super accuracy model. What this means is that the raw data is processed by a tensor, like artificial intelligence algorithm that detects the current through pores, and from the current, it identifies the base. What is good is that it's actually very appreciable. Also, now Singleron bioinformatician team produced a next flow pipeline. It means that users of long-read sequencing can use Epitomy Lab. This is the next flow platform for long-read sequencing. And they can import this pipeline and directly analyze their data. So from FASTQ files from Dorado, they'll get a matrix file. Actually, all the files that a 10x system will produce. So they can go further to Surat or Monocle. This is for the audience that is actually using more or less single cell sequencing. So in this data set, we did barcode correction. I have to repeat a little bit so you can get some understanding. So barcoding of cells is very important because by this barcoding, you identify each cell from the others, because when the cells leave the chip, you lyse them first, you amplify the mRNAs there, and then all those mRNAs, they'll be put together. So in order to distinguish what is inside, you need to identify each cell, single cell, by its barcode, and each molecule by a unique molecular identifier, UMI. So these UMIs actually, they're very important because then in single cell sequencing, you don't have expression like in bulk sequencing, you have counts. And counts you analyze counts by counting the UMIs in each cell. So if all barcodes, if they're equal, then you have one cell. And then the algorithm calculates how many UMIs you have. If they're similar, then all is pulled together. This is one, these are pieces from one single RNA. If you have multiple same type RNAs with different UMIs, then you have several molecules. This is how it's working, because this is very complex matter actually, because this is a sparse matrix. It means you have a lot of zeros, a lot of undetected RNAs, and only few are detected. Based on bulk sequencing and mathematical modeling, you can actually have a picture of the expression of single cell, because as I'll explain to you, a lot of, per single cell, you can have like 1,000, 2,000 genes, but you do not have like 20,000 like in bulk seq. But you have specific expression for this cell. So we selected all RNAs that all cells that have at least 200 genes detected. And for each gene, it should have been expressed at least in 3 cells. And we use the standard for 20% mitochondrial genes in a cell. So otherwise, we filter. What you see here is a long-read sequencing quality analysis. The median of the sequence data is more than Q20. It means 99% accuracy or more. The average length of detected reads was like 900 to 1.2K nucleotides with N50 of 5 kilobase. It means that the half of the library is the molecules inside are at least 5 kb long, which is enough to detect most of the full-length RNAs. 70% of the RNAs were detected and mapped to the GFF transcriptome. And what you see here on the left is the white type line. On the right is the carcinoma cell, semi-rich cell line. So they overlap, but they're not identical. This is produced by this workflow I showed you. After that, we do bioinformatics with other programmatic means. So in average, to summarize, this very early version of the kit produced that was able to detect about 104,000 cells that have all barcoding, UMI, and full-length transcripts after filtering. When we use the bigger flow cells of the Oxford Nanopore, we can get up to 5,000 cells and up to 2,000 genes per cell, which is, I think, a very good start for. We haven't tested actually your HD new chips, so it's about to. We can share this information later when we test. This is with very early version. 98% of the mRNAs detected actually maps to full-length RNAs, which means that the barcoding technology is, and actually the barcoding technology is very good because it can capture the whole transcriptome efficiently. So these are gene-level UMaps. They show, as I explained to you, you have a multidimensional matrix per each cell. So for each cell, you have a number of the cell, like the barcode, and then you have different genes with their expression levels. So what you get is that you need to make this information of 2D in order for a human to comprehend. So what we see here on the left is the two population. The orange one is the cancer semi-rich cell population. There are 3 clusters. And as you see in orange on the second figure, not all of this population of cells, it remains specific only to the wild phenotype. When you induce most stem phenotype, you don't have it. So you lose some of the expression. Then we have these 2 figures show that there is no separate cluster of cells that will carry the most expressed genes. It means that technically there is no artifacts in this data set. Similarly, when you use the advantage of long reads, you have transcripts directly. And as you see on the first graph with orange, you have the stem cell population. We have 12 clusters. And the good thing of a single cell technology is precisely that you can have statistics on each cluster and separate expression for each cluster or for each cell. So similar to bulk-seq, you have a differential gene expression. You can identify genes that are most abundant. As you see, genes that were most abundant here they're actually not specific genes that are overexpressed. There are a lot of genes that get changes in their expression after acquiring this new phenotype. Between 60 to 80% of the cell population actually bears these changes, so they're coordinated. And they are related to interferon stress signaling, secretory and plasmic reticulum, and lysosomal remodeling, and also MLA-T1 long non-codec RNA was overexpressed after we induce this change. So when we get to transcriptomics part, we identify that at least the medium of the gene, the majority of the genes had 1 to 2 transcripts per cell. Initially, back in the years, the research community believed that one cell produced 1 transcript. But now it's known because due to these technologies that one cell can produce, as you see, up to 11 transcripts. Majority of the genes, though, they expressed like 2 transcripts or more. 38% have expression than more than 2 transcripts per cell, which is very interesting, not only biologically, but also if you seek for targets. And what I'll try to show you that this technology will actually reveal enough information for precise targeting of cell. So as you see here, the pathways that are affected by these most transcript abundant genes are seen. And we have we identified more than 4,000 genes that have more than 2 transcripts, which are interesting for the further analysis. So what we can do with this information? We have differential gene expression per transcript. This gives you information which transcripts directly are, how abundant they are. And you can see this, we can visualize this with UMAP. So we can see these are the most abundant transcript, and what I put in red square. This is the region for the cancer stem cell population. Most of them are upregulated. Some of them are retained to the adherent phenotype. As you can see here, the transcript from the blobs, we can actually have a good visualization of the expression level and the abundance of the cells. The size of the blobs reflects the percentage of the population. So we see a coordinated change after we change the phenotype. Even more interesting is when we get to the differential transcript utilization, which you cannot do with other. If you do only 3' single cell, you cannot detect which transcripts are used. And on the figure on the right, the size is based on the expression that you saw before. The color is based on the number of transcripts used per each gene transcripts. It means that, for example, the red dots, all of their transcripts up to 100% are changed between the 2 states, while the genes that are very abundantly differentially expressed actually have less changes in the transcript. Like this gene TMM2P58, it's a transmembrane protein. As you can see, when you visualize the reads, in the wild form, we have 2 with green arrows I pointed. These 2 transcripts are retained in the cancerous phenotype, but there is a novel one with intron inside, and there is one point that probably this cryptic exon, because it is in the intron part. It shouldn't be there. And you see multiple reads that confirm that. And this is not deep sequence. This is a small fold cell. So usually deep sequencing gives better result. And as you can see from the graphs, on the left, we have leader switches. Leader switches, there is a specific statistic that allows you for each transcript, for each gene, actually, to see how different transcript it encodes, what ratio they have in the transcriptome. And what we see on the left is that these 2 genes, the one I just showed you and one other UCML1, they both change entirely from the one phenotype to the other. So when we induce this cancer stems phenotype, the expressed transcript is totally abrupt, and another transcript is has been started to be produced, to be transcribed. The other, though, they have only rewiring. It means that only the expression is changed, or new transcripts are started to express, but the transcripts that were in the original phenotype haven't been fully depressed. So if we want to target something that is very specific for the 2 states, we could get those that are so-called leader switches. They just switch on off. And you can see this, and you can have this kind of information for every single differentially used transcript. So you have expression and utilization. And the last thing I want to show you, this is a little bit more tricky to use and interpret. This is a kind of trajectory analysis. The trajectory analysis, there are a couple of ways to do, but the whole idea behind is that you want to see if from one state to another state, there is a set of genes that will guide you from one state to the other, and you align those patterns between these cells on a pseudo timeline. It means that on this third figure, this x-axis provides the timeline, and the y-axis actually gives you changes in cells. Between these two states, there is not abrupt transition in the whole pattern of genes. When we interrogate for like epithelial state, mesenchymal state, proliferative state, also stemness, there are several genes that can make few or more. So in this gene set, we have, for example, epithelial cells 5, mesenchymal cells 4 out of 7. But the statistics, there is minimal correlation between the change of state. So we have genes that we detect, they belong to epithelial or mesenchymal phenotype. And we know that this is already a cell line that has undergone epithelial to mesenchymal transition for some part. So we further push this cell line to be even more metastatic, but there is no statistically significant change in this case from one state to another. Sometimes you can have in a big trajectory analysis different states, so you can see, for example, from stem cell to differentiated cell, there is big difference. What you see here is vimentin and one org gene. They have slight transition. They have some statistics that shows that between one of the states to when they're getting this STEM's phenotype, STEM-enriched phenotype, they have some changes in the expression, but it's not very diverse. What we see from this analysis, actually the switch in the transcripts is more specific for these two states rather than this transitional state. And actually this allows us to select targets based on the leader switches rather than the trajectories, which are method to actually use differentiation. So with this, I would like to thank my colleagues. That's not the last version, yeah, for helping me arrange this local part of the presentation. And once again, to tell Singleron and thanks to Namfang to give us this wonderful lecture. I hope more of you will be excited as we are by the colorful world of single cells. Thank you.
So if you want to ask some questions to the professor.
Or if you first get that, yes, yeah.
Yeah, thank you very much. Although I know you have been doing all the work, but it's the first time I really saw the complete presentation of results. I'm just wondering, it's really very striking to see like the different isoform expression patterns, like in the cancer stem cell status compared to ADH. What do you think could be the higher level mechanism to regulate that?
Well, because we have bulk sequence of this, and what we actually do in this project, we try to combine cDNA, direct RNA changes, epitranscriptomics, plus single cell. We want to see from different parts what happens when we make this, because the phenotype in the beginning, as you see it from adherent, they become organoid-like. There are I don't know if I showed or mentioned, but there are some epigenetic chromatin organizers that are changed actually. So we believe also there are genes related to spliceosomes. There are genes related to ribosomal complex RNA processing, and a lot. These are all long non-coding RNA. So we believe this change is not actually artifact phenomenon, but it's rather when they lose their ability to attach to a surface or like in tissue, they start to grow attaching to themselves in organoids. And this phenotype change is probably related to this change in RNAs in this switch. Why specifically this gene, one of the gene related here, the TM, sorry. No. So this TME, this is transmembrane protein, but actually it's involved in cellular trafficking and endoplasmic reticulum functioning. So I believe the new biology they acquire that cannot attach is related to these changes in because this leader switch changes. It means that the function of the RNA that is used is different. Like in some genes encode several isoforms, and some of them are regulatory, some of them are functional. Including, for example, because we investigated for a long time in inflammasomes, the scaffold protein of the inflammasome it is a effector molecule, but actually it has a truncated form that is regulatory, and when it's produced, it has negative regulative feedback in the nuclei. So I cannot say for this, we haven't studied them functionally, what is their specific function, but we believe there's a complex regulation because the data shows coordinated change, so this phenotype is acquired. It is selective pressure because we change the media, we change the simplest protocol is a serial trypsinization. But also we put them in a condition that stimulates stem cell development. So if I have to be correct, these are more or less cells acquiring stem-like phenotype. It's not genuine stem cells. Probably if we compare even with cancer stem cells, that they would have broadly, distinctly different transcriptome also. This is what I can tell at this stage. Thank you. Thank you. So